to be attending today's talk, and I'm very delighted to be part of this great consortium, and thankful to Rebecca and others, many other people, to have put this effort, this consortium together. It's a pleasure to speak about the TIL trial we're about to launch. First, to me, a T cell is a very concrete thing, and I hope it's going to become very concrete to you too. When you see what a T cell is capable of doing, if I can get this video started here. What's going on? Huh? Yeah, I'm on the arrow, but it seems it's not. Sure. Oh, here it goes. Okay, so that's a T cell that is going to attack a cancer cell. That's in a dish, it's in the laboratory, but the same thing can happen in your body. And it's really the role of T cell. We have 1 billion T cell per ml of blood that are continuously patrolling the body and looking at the surface of all cells in our body, which one has an abnormal peptide. And when the T cell finds an abnormal peptide, it can destroy this cell, this cancer cell. So this is very concrete and inspiring. T cells, what we think best recognize and enables destruction of cancer cell is called neoantigens. That's a very important concept. It's a simple concept. Cancer cells are mutated. The genes in cancer cells contains mutation, and those genes are expressed, translated into proteins, and those mutated proteins are chopped into small pieces that we call peptides. And those mutated small peptides are brought at the surface of cancer cells. And these are the neoantigens that can be recognized as foreign by T cells patrolling the body. The good news is also that we have a tremendous diversity of T cells bearing different receptors that can recognize many neoantigens. And the other good news is that those T cells that patrol the body have been educated not to recognize our own antigens. And so they don't attack the self, and TIL generally does not cause autoimmunity. The TIL therapy that you've started to heard about is also quite concrete and simple. It has been designed by surgeons, so it has to be simple. You the surgeon extract a metastasis, does a surgical resection of a metastasis, and this tumor tissue is chopped into small pieces, put in very good media, enriched with all sorts of good elements that T cells like, and the T cells proliferate to very large number in the incubator. And this becomes a white blood cell product, transfusion product, that is going to be reinfused to the patient from whom we resected the metastases. For this therapy to work, the patient needs a lymphodepletion, an immune preparatory regimen. This lymphodepletion creates space for the T cells we're infusing to work, and it momentarily reduces the immune system defense of the host to allow space for the new T cells to work. And it's about a week of immune suppression that is mediated by the lymphodepletion. And during that window of time, we also give patients some immune stimulants, IL-2, that stimulates the T cell product we've infused to proliferate in the body of patients and kill as many cancer cells as possible. The overall recovery in the hospital is usually 7 to 10 days after the infusion of the TIL product. So this method, this TIL therapy, has been developed mainly for melanoma, skin cancer. Skin cancer is the cancer best recognized by the immune system. And you can appreciate here slides from the NCI from Dr. Rosenberg's, who is really the pioneer and the father of this form of therapy, showing here a patient 25 days before TIL with a liver that is filled with metastases. Those spots, darker spots, are metastases. And you can see after a month after TIL therapy and 5 years after TIL therapy, this patient enjoys a complete response and has no more metastases. So bulk TIL like that works nice for melanoma. It is now approved by the FDA in the United States for use as standard of care and in some European countries. So the real question for us is, can TIL work for cholangiocarcinoma? And we'll make the argument that it can work. And we have the living proof of that here, Melinda, who's gonna follow up with, yeah, she's gonna follow up explaining what is TIL from her perspective. I was fortunate to be trained in Dr. Rosenberg's lab around 2012. We launched a first TIL trial for gastrointestinal cancer. And in this trial, we could recruit patients like Melinda with cholangiocarcinoma. And we learned a tremendous amount of information from Melinda's journey. We've expanded a bulk, a conventional TIL product that, as you can see here, after infusion, mediates some regression in the size of her metastases, but then the cancer came back and grew quite significantly. And we were then able to generate a TIL product that could attack more specifically, that was highly enriched in attacking neoantigens. And Melinda enjoyed a much deeper response to that second infusion. And combined with some immune modulator, an immune checkpoint inhibitor you may heard of, anti-PD-1 drug, and other components of her treatments that Melinda's gonna talk about. Well, she's here with us today, almost 15 years after treatment. So TIL therapy can work for those less immunogenic cancer, but we need to enrich those T cell product in T cells that are capable of targeting neoantigens, and we need to combine with other immune therapy for this to work. So you may say, well, okay, this worked for Melinda, but can it work for other patients with GI cancer? The surgery branch has just published, like last week, this week, the summary of their of the entire GI TIL protocol. It's on roughly 80 patients or so. When we started that protocol at the NCI, we were just giving TIL like for melanoma, and you see the response rate was 0%. Then we developed technique to enrich the TIL product in TIL reactive to neoantigens, and the response rates got up a little bit. And then like for Melinda, we did not only enrichment and select TIL that recognize neoantigens, but combine this with the immune checkpoint targeting PD-1. And the response rates reported now is about 23.5%. In all those patients treated, only 6 had cholangiocarcinomas, and 2 had objective responses, and Melinda is one of the 2. So TIL can work for cholangiocarcinoma when we select, when we combine, but how often can we find neoantigen reactive TIL in bile duct cancer? The best data available was published again by the Rosenberg group in 12 cholangiocarcinoma. And it seems that for the majority of those cancers, we can find some T cells that recognize neoantigens. Okay, so what to do with that? What could be the best design for TIL for cholangiocarcinoma? Most of you are aware, you're cholangiocarcinoma patients, that the first line of care is gemcitabine, cisplatin plus anti-PD-L1 therapy, durvalumab. This treatment is more effective than chemotherapy alone, and certainly more effective than no treatment at all. But you can see by the progression free survival curves here that in 95+% of patients, cholangiocarcinoma is going to progress despite GemCis and durvalumab. But this approach is interesting because it tells us that giving immunotherapy, combining immunotherapy with chemotherapy for this cancer, stimulating the immune system for this cancer has a benefit. And this is what we want to amplify. We want to give as many T cells, like the one I showed you, able to destroy cancer cell, as a T cell product to really boost that immunotherapeutic component of the therapy. So how are we gonna, what kind of T cell product we are going to make? We want to give as many cells as possible, and we want to give as many cells that recognize neoantigens. And we've developed two techniques to enrich in tumor reactive, neoantigen reactive T cell. The first one is called the antigen agnostic approach. It's based on T cell biology. A T cell that has repeatedly seen a neoantigen presented by the cancer cell is going to express at its surface a molecule called PD-1. That's the exact molecule that anti-PD-1 drugs is targeting, but we use PD-1 as a target to sort tumor reactive T cells. We don't care which antigen is recognized. We have this as an indicator of recognition of any neoantigens, and so we sort the cells and make one bag for infusion with this technique. The second bag we're making to infuse to patient is based on direct neoantigen recognition. We can now sequence tumor, predict neoantigens, and we can assay in the manufacturing process, we can assay neoantigen recognition and select the reactive TIL, make a infusion bag from those neoantigen reactive selected T cells. So I will invite Sandy Pelletier and Mathieu Crupi to continue this talk to explain a little bit in more detail what is this manufacturing process, and then I will come back to talk to you about the overall design of that trial to close. Sandy is really an expert in adoptive cell therapy. She's working with us at the CHUM. She is leading really the manufacturing at our team, at our site. She has years of experience in generating TILs in multiple cancer types from patients' tumor samples. Mathieu Crupi leads the Personalized Biotherapeutics Division at the Ottawa Hospital Research Institute, the BMC facility. He's also working on TIL manufacturing, CAR T cell manufacturing for upcoming trials. So please, Sandy and Mathieu, come to the podium. We'll take questions at the very end.
Okay, so I will not go through all of this slide. For those of you who are wondering why hasn't the trial started yet, this is why, because it's a very complicated way of making TILs, like Simon mentioned. We're not just isolating all of the TILs from the tumor, and we're not just all infusing them. We want to select the best ones, the ones that could really kill your tumor. So basically, these are the two methods that we use: the agnostic approach, where we use a cool cell sorter to sort the cells that express PD-1. And then with the same tumor, so basically we just split the tumor in half when we get it. We take half the tumor for part 1, half the tumor for part 2, where we grow the different fragments in multiple wells and try to enrich them in tumor-specific cells. So just to give you an idea of the work that it takes. So this is the number of procedures, so the number of protocols that we actually have to write to manufacture these TILs. So we have, several dozens of protocols for, the cell production, the recognition assay of the tumor, quality assurance, quality control. So we need to make sure that the cells that we're reinfusing to you are safe, that we haven't introduced any contamination, and that, the cells that we are giving you will be healthy and active. So this is just one slide showing some of our results. So this is the number of PD-1 sorted cells. So sometimes we start with very low numbers of cells, like 1,000 cells. And after 14 days in culture, we're able to get 10 to the 9 cells. And this is with like small pieces of tissue that the pathology gives to us. And even with that small amount of tissue, we're able to grow. And this slide is with melanoma tissue. And then we have all of these different samples from GI cancers. So here we can see the fold expansion. So even for melanoma and GI, we're able to get close to 10,000 fold expansion. And here we're just showing purity. So that's very important for Health Canada. We need to show that we're only giving you CD3 positive cells, which are the T lymphocytes. We don't want to give you anything else that was isolated from the tumor. Here is also another slide. I'm not going to go through everything, but this on top is flow cytometry to show that we're able to detect neoantigen reactive TILs. So here visually, maybe we could just look at this plot. So basically CD137 and CD134 are like activation markers. So we show that if we put the TILs with the mutated peptides, we see all of these cells that start to express CD137. So we're able to grow these TILs. So that's very encouraging. I'll just talk about our clean room. So it was built in 2017. It is within the operating room of the hospital. It's meant for cell culture of clinical samples only. We have a quality system. We have monitoring 24 hours for all of the equipment. It's a room with positive pressure to make sure that there are no contaminants that are entering the clean room. We have a team of 7 highly qualified staff. This is us just last week with Cecile, who's here, and Katia and Elodie, where we're going in the clean room doing like this big experiment. So as you can see with the smile on our faces, we're very excited to do this work. So Mathieu, your turn.
Hi, everyone. So in Ottawa, we're also very passionate about the work that we're doing, and we're so lucky to have great collaborators like Sandy and Simon that have made the tech transfer possible for TIL work, and I'll get into the details of that in a second. But to start, I work at the Biotherapeutics Manufacturing Center inside of OHRI. So if ever you're visiting Ottawa and you'd like a tour, I know there are a lot of patients and advocates here, we'd be happy to facilitate that. And our director is here, Dr. Jennifer Queasy. If you want to put your hand up, putting you on the spot, but we'll make sure that can happen for you. And so at the BMC, we've traditionally manufactured a lot of different viruses, oncolytic viruses. These are cancer-fighting viruses. Some of those have been, initiated and designed by Dr. John Bell, also sitting next to Jen Queasy over there. John, if you want to wave. And so we're very collaborative. We work a lot together. And the idea here is we want to be able to manufacture these products for Canadians. So as we've been doing for the viruses, now we've been going to the direction of also doing this for many types of cell therapies for cancer patients too. So as was mentioned, we have a CAR T program, but more recently we've been developing TIL manufacturing with our collaborators. And so this is a picture here of our team. We're about 40 employees. We've already manufactured many viruses and different cell products for at least 15 clinical trials, and these have been in Europe and Asia and Canada for phase 1 to 3 clinical trials. So it's very exciting work that we do. It's a privilege to work at the BMC. And this is the new suite that we recently designed in the past 2 years since C3 started to be able to allow us to do the same thing that Simon's group is doing at CHUM. So we could be a second manufacturing site. And the technology process that I was telling you about that transfer is really a transfer of knowledge. So they will give us standard operating procedure documents. So these are SOPs for short. Basically, it's a lot of paperwork that tells you exactly how to do these experiments, how to grow tills. And so we've been able to start doing that process in-house with our team, and we've been able to grow up tills from these fragments, just like Sandy. And now we've even done a demo for the title, the instrument, the flow cytometer that she talked about, to show that we could sort things and demonstrate comparability between our two sites. So this is going to be important moving forward because eventually we're going to do a clinical trial. We want to make sure that both sites would manufacture the same product at the end. And so this is an image of the room that we've built out. You can see there's an isolator here. It's like a glove compartment box, but it allows us to work in a very sterile environment to ensure our products are safe. Here's a picture of our team. We also work on process innovation, so try to make sure that we can make this process even faster for patients and more cost effective, which is very important in Canada when we have a public healthcare system, as you all know. And my last slide here, I just wanted to mention that part of releasing these products, as was mentioned, is making sure that they are safe. So we have different release assays that we develop in-house. And luckily, a lot of these are similar for CAR-T as they are for TILs. And we have different pieces of equipment like this Bactilert machine, which is a giant machine that allows us to make sure that there is no bacteria or fungus in the samples, for example. And so I think I'll end with that. Thank you very much, and happy to answer questions at the end.
So we've shown you why we're doing a sophisticated TIL product, but you're probably wondering, well, if it takes 3 months to do a TIL, how are you going to fit this into a clinical trial to treat metastatic cholangiocarcinoma? So I'm going to close on discussing the trial design. This was the synopsis was developed during multiple rounds of consultation with experts in Canada and international experts in the TIL field as well. We and patients. We would like to recruit patients with metastatic or non-resectable but metastatic to lymph node cholangiocarcinoma, bile duct cancer. There needs to be a metastasis that is surgically resectable with low risk, right? If there's no tissue we can harvest, we cannot make a TIL product. And in some case, it's for cholangiocarcinoma, tumors can be quite central in the liver, they can be infiltrative, and it's this criteria is unfortunately one that can limit the eligibility of patients with cholangiocarcinoma. There needs to be some measurable disease also, so we can make sure that the treatment is going to be, we can measure if the treatment is effective at the end. One needs to do not benefit from the standard of care, Gemcis, Durva, the first-line standard of care. We cannot ethically propose to our boards to treat with in a phase 1 trial, first in human type of trial, a very experimental treatment. And so this is something that needs to be in the inclusion criteria. There also needs to be no dysfunction of the bile flow. So a lot of you know that those cancer, since they arise in bile ducts, they can block the ducts, and many patients have stents to open these ducts, and it makes patients susceptible to infections. One cannot enter lymphodepletion, this window of 1 week without their immune defense, with an ongoing infection in the liver due to obstruction of bile ducts. And this is a very important criteria for that study for the safety of patients. Most of the toxicities and the severe toxicities of TIL treatment relates to infection. And so that also for cholangiocarcinoma has specific implication because of the bile duct issues. We want to recruit 15 patients, and this is going to be a 2-site two sites study. It's going to be running at the CHUM and in Ottawa. So patients can come to Ottawa, have their surgery for tumor resection. The tumor is shipped to Montreal, to the CHUM that initiates their manufacturing. When Ottawa is going to be manufacturing, the cells will be manufactured in Ottawa too. And when the time comes for treatment, patients that enrolled in Ottawa are treated in Ottawa. They receive their cells there, and Quebecers receive their cells at the CHUM. So the idea is to enroll patients as early as possible in their journey. One is diagnosed with metastatic cholangiocarcinoma. You can be recruited in that trial, and we want to schedule surgical harvest of the tumor and what we call leukapheresis to get antigen-presenting cells for the manufacturing process as early as possible in the treatment scheme. So then we'll have time, we'll have that 3 months to produce a TIL product. And it's only when one is not having any benefits from gemcis durva, progression, toxicities, potentially other reasons, that then this triggers the treatment phase. We reassess, we reconsent the patients that were first enrolled to make sure they don't have any active infection, they're fit for TIL infusion, and if this consent is a go, then we do the treatment phase, lymphodepletion, TIL, IL-2, and the maintenance anti-PD-L1. This maintenance anti-PD-L1 is good for TIL therapy. I showed you in the first slides, we need to combine targeting PD-1 and selected TIL. Then the assessment period starts after TIL treatment. It sort of overlaps the TIL treatment period because of the anti-PD-L1 maintenance. And we've set as primary endpoint safety and tolerability, TIL manufacturing feasibility, and as secondary endpoint efficacy. And we have, this is within C3, you've heard about the research. There are many exploratory objectives to try to understand why is this therapy working in some patients and why other patients do not respond to this treatment. There's going to be a lot of biospecimen collection and harvest at multiple times during this trial to best study the mechanism of action. Before I close, I want also to mention that we will have philanthropic funding to open a cohort of patients that's going to be a cohort for all comers. So I'm really attached to this idea that if one patient is fit for TIL but doesn't meet all those specific criteria or has survived gemcis durva in a targeted therapy and could be a good candidate for TIL, well, let's try this approach in those patients as well. So there's going to be also this possibility in that trial. So we've completed the preclinical trial application to Health Canada, where we presented the synopsis, the trial design, and the manufacturing process, and this was successful. We've secured, there are many steps and hurdles we're not presenting here, but I want to highlight one. We need human albumin to produce our cells, and this is a highly controlled blood transfusion component, and it's administered by the well, in Quebec it's called Héma-Québec. In Canada it's in the rest of Canada, it's the blood services, right? So it's controlled by this entity. And we needed the Ministry of Health authorization through Héma-Québec to get albumin to do our manufacturing. And this took 6 months of back and forth between our lawyer's office. And so that's one example of everything that needs to be put together for doing the manufacturing. Currently, we're aiming to do the engineering runs, the pilot runs this fall. We're working very hard. Sandy and Cécile, who's here, working on the last pieces of processes development for release criteria testing. Mycoplasm is a microorganism that we also need to make sure is not in the cell product, and some very fine tuning on the cryopreservation step. In the fall, it's going to be a very busy fall. We are thus going to do the pilot manufacturing runs, the engineering runs. We need to secure the anti-PD-L1 drug for the trial. I don't know if anyone from AstraZeneca is in the room today, but you will soon hear about us. So we need to develop the full protocol and related manuals, consent form in the Ottawa and CHUM format, the trial monitoring and data plan. We need to finalize contracts between our institutions to run this trial. We need to do the full application to Health Canada, the clinical trial application, and get our clinical research unit to operationalize the trial execution. So with this, I want to acknowledge all of the people in my lab, so the biobanks that is supporting this effort at the CHUM, the pathology, soutien de la recherche, the surgical colleagues who are contributing to this, our funders, everybody from the C3 have been also involved in this, and I welcome any question. Sandy, you can come back.
Thank you so much for your talk. I have two questions. Number one, do you think that there is any predictive biomarker for that trifecta treatment, 2 chemotherapies and that PD-1 therapy? And number two, do you think, have you ever thought about patients getting full remission under that therapy? So if it is worth spending the time and money to expand those TILs, and if there's no hope of ever getting full remission with those 3 drugs, do you think there is value to store them, or?
So repeat the second question is?
So the second question is that when it comes to developing resistance and non-responsiveness to that 3 drug,
Yeah.
is it a matter of when or a matter of if? And if it is a matter of when, how long would it be, and how do you envision storing those cell therapeutics that you've developed?
Okay, so I'll start with the second question. So unfortunately, 85–90% of patients who receives GemCis plus Durva progresses over time. And you see a year after initiation of the treatment, about 80% of patients have progressed. So that's why we want to recruit, harvest tumors, start the TIL manufacturing as early as possible after patient diagnosis. And we do, to make it simple, half of the manufacturing, and we cryopreserve, we freeze TIL. And then we wait for any signs of progression, radiologic progression, clinical progression, raise in tumor markers like CA19-9 or toxicities. Patients cannot tolerate the chemotherapy. This triggers the TIL treatment component. We finish the 14 days manufacturing we need, and we can infuse the T cells.
It is so the probability of them developing resistance is very high, so it makes sense to develop those cell banks.
Yeah. Okay. Yeah. For biomarkers, Melinda wants to. Yeah. Yeah. Right. When we went over this in synopsis development, and most oncologists and most institutional ethics review board are not accepting to offer a highly experimental treatment before a proven standard of care. So it's very difficult to offer TIL prior to doing a first shot at a standard of care. Yeah, for biomarkers, so this science is evolving very quickly. Many patients with melanoma have been treated with TIL, and accumulating numbers of patients with other cancers. But currently, there's no great biomarker to distinguish who's going to respond to TIL and who's going to not respond. It's multifactorial. It's not only the initial tumor, it's the host also, the microbiome, the TIL product. When we infuse those TIL, some products contains T cells that are end of life and about to die. Some other T cells has an extraordinary capacity to renew in the body, and that's something that is not yet controlled in the manufacturing process. So it's a complex field, biomarker fields for TIL. It's evolving quickly, and at one point we'll have those biomarkers. For these three drugs, there isn't a great biomarker. For GEMS's, to predict GEMS's Durval response, there is no biomarker. Yeah.
Biopsy the metastases. Oh, sorry.
It's on.
When you said biopsy the metastases, do I understand you to mean that you could biopsy, for example, lung nodules?
Yeah.
You could, and
Yeah.
that would give you. And the second question is, when you talked about a wider demographic at some point, that you're very much in favor of a wider demographic, does that mean that you're in favor of lowering the exclusion criteria? For example, my wife we're both towards our 80s. Many trials would exclude us. She also has reduced EGFR in her kidneys. Would eventually this treatment be available to people like us?
Right. So this, the trial design I've showed you, this is one arm, very strict upfront till as early as possible. That's one arm. But we want to open another arm that's going to be for all comers, and not only cholangiocarcinoma. And we're making sure in our detailed inclusion criteria that we open possibilities to as many patients as possible. For instance, it's up to 70 years old, but above 70, it's on a case-by-case basis. So many patients has 80 and chronologically wise, but biologically wise, they can be much younger and fit. And so we could recruit patients that are older than 70 years old.
I had a couple of comments. Nice work. I had a couple of questions, but comments. I mean, regarding the first line versus second line, this is a huge problem in immunotherapy, not only for cholangiocarcinoma, but other cancers where we're treating patients upon progression of checkpoint blockade. And, when I was at MD Anderson, I grew a lot of TIL from different types of cancers, including melanoma. And unfortunately, even growing out the TILs from a patient that has relapse after checkpoint blockade is a lot tougher. You don't get enough T cells, you get less robust T cells. So the whole field is really pushing in this area towards this type of T cell therapy, but more frontline and earlier lines. And I think you can have your cake and eat it too. I mean, this is an amazing first step, because if you prove that you can do TIL therapy reproducibly in these patients after recurrence or failure of DARVA and chemotherapy, you can then set the stage to do a randomized trial of DARVA plus TIL versus the standard of care. And then if you can do that, then you're platinum. And I think this is the key, setting the stage to do that and getting to that point as soon as possible. Because what we're finding now in neoadjuvant and, first-line therapy, for example, Iovance is doing in melanoma, with PD-1, they're seeing much higher response rates. And this is where you're going to really improve patient survival and treating healthier patients that haven't been treated with multiple lines of therapy. And I think it's a huge misnomer, and I'm sorry if I'm going to say something controversial because I've been in this field for many years and grew TIL. Trying to grow TIL from patients in melanoma is hard enough that have failed checkpoint blockade, yet alone these other types of cancers. So I think we really, as a community and patients, need to advocate for getting these therapies more frontline and having access to these and getting these as a standard of care. My second question is a science question, or my question is a science question. Given our data too that NAGME showed too as well regarding shedding of HLA class 1, I think are you guys, well, I guess it's a question for, I think, are you looking for CD4? And so there's two types of antigens that present tumor antigens on the surface. There's class 1 MHC and class 2 MHC. The class 1 MHC activates what is called the cytotoxic CD8 T cells, and the class 2 MHC presents antigens to what are called CD4 helper cells, but those helper cells produce cytokines like interferon gamma and other things that can help kill the tumor and tumor necrosis factor. And so generally, immunotherapy has been really harking just on the CD8 class 1. And we haven't paid attention. And I think Melinda will attest to it, right? Because your cells were CD4 T cells, right? So my question is, are you guys looking at CD4 neoantigens and tracking those?
A lot.
And what?
So in the last paper I quoted that summarized the GI TIL experience, the Nature Medicine that just came out this week, the actual response is greater in patients that had CD4 reactive neoantigen TIL. And that's what we see also in our screening assay. We didn't go into the detail, but after primary expansion of the TIL, we test the reactivity of all TIL subcultures to neoantigens. And what are these neoantigens? We use long peptides, 25 mers pulsed on autologous APC presented to the TIL. And we typically will detect much more CD4 reactivity than CD8s. And we select the subcultures that are most enriched in reactivity, regardless if it's CD4 or CD8, but we do find a lot of CD4. So the end product is generally enriched in CD4 T cells.
I want to sneak in with one question related a bit to Laszlo's first question. Do you think if the trial is open and a patient is already, let's say, on Gemcis Durva, or they finished their Gemcis and they're on their maintenance Durva, could they get a harvest at that point if they still have a resectable tumor? I mean, obviously we'd have to do, they're going to have a different TIL product, but that was the question. Could they get harvest after they start?
Right. I think definitely this consent one for tumor harvest can really happen anytime we think is fit and the oncologist and the patient think it's the appropriate time. Like the bias is to do it as early as possible because it's going to give us a greater opportunity to treat more patients. But someone stable on PD-L1 therapy, we don't even need to stop the durvalumab, and we can readily proceed to tumor harvest and leukapheresis and just wait for that time where the cancer starts to pick up to infuse TIL.
So just to that point, getting access to the clinical trial. So as and when this becomes available as a clinical trial for patients that are either just starting on their journey or they've failed the first line of treatment, how do they advocate for themselves to have this be considered with their oncologist? Is the expectation that we would have to be the advocate ourselves and bring it to the oncologists? Or is the expectation that the knowledge would be shared so that the oncologist would know to recommend that?
Right. There's no expectation, and it's always a mix of both, right? We're once we are ready and we open the trial, we'll let it know to as many oncologists as possible through the C3 network and our rounds, and we'll do as much advertisement as we can. But then I think patients foundation like the Cholangiocarcinoma Foundation can also relay the information, and then by word to mouth at some point, patients that are diagnosed with cholangiocarcinoma will go online and look at what are the resources, and they can talk to their oncologist, And that's how we can do it. There's no way we can inform each and every oncologist or each and every patient, but we can advertise as broad as possible through as many channels as possible.
Well, hi, thank you. The first step in the TIL therapy process is depletion or lymphodepletion.
Yeah.
And is that complete depletion like what you would get if you were to have a stem cell transplant?
So it's pretty much along a stem cell transplant, but this lymphodepletion is milder, and it doesn't kill the precursor cells. So for a stem cell transplant, you want to replace completely the immune system of a patient. For this, you just want to do enough lymphodepletion to create space, and then the own immune system all comes back, but it's skewed with the new cells that were given.
Okay, and the drugs that are used to do this depletion, could they also potentially help other cancers to develop?
It's cyclophosphamide in English, and fludarabine. These drugs are used for breast cancer. Cyclophosphamide is. The typical dose that is given is more geared towards creating the lymphodepletion, but it's not excluded that it can also stop fast dividing cancer cells. So it can contribute in this way as well to the treatment scheme.
Okay.
It's not conventionally used to treat cholangiocarcinoma,
No.
those drugs. Okay, but there's always a possibility that a cancer drug, that mechanism of action is to target a cell that is in fast division, can stop any cancer cell.
And you were saying that the first line of treatment for cholangiocarcinoma are the three drugs.
Gemcitabine.
Yeah, right. And you said that it was a proven therapy, yet it seems that it only works in 15% of the cases, because you said 85% after a year
Progress.
will progress.
Mm-hmm.
So it's a pretty, when you have something like a disease like
Yeah.
this, you want the best.
Right. And so that's what we face. That's why we're here. We need to do better than that. That's what's approved, and it's not great. So we need to do better.
Thank you.
Yeah, I would, I'm just gonna put the slides at the right place. I would like to introduce you to Melinda Bachini. She is a 15-year cholangiocarcinoma survivor who was treated with TIL in 2012 in a clinical trial at the NCI. She's now the Chief Patient Officer for the Cholangiocarcinoma Foundation. She leverages her experiences and passion to support those diagnosed with this disease. Welcome, Melinda.
I just want to echo, oh shoot, I don't have to touch this, do I? I just want to echo all these comments, because this is something I've been talking about for years of, I would so rather see patients go for the home run in the beginning, because if it doesn't work, chemo's not going anywhere. Gemcitabine is still there and we can still try it. I just want that opportunity while we're still healthy and doing well to have that shot at being here 15 years later. I'm Melinda Bachini. I'm from Billings, Montana. Thank you for welcoming me into your beautiful country. I may not go back. I feel like I need to get a shirt that says I did not vote for Trump when I travel out of country. I just am briefly going to share my experience with doing the TIL therapy and put myself back into the position that you guys are all in. And I know after hearing Cynthia and Dana and all of you guys talk to each other yesterday, the one day that we'll never forget is the day that we heard those three words, "You have cancer." And I will never, ever forget. I remember where I was standing in my home. My kids had gone to school. My husband had just gone to work, and the phone rang, and it was my gastroenterologist. And she said, "I'm so sorry, it wasn't on my radar, but you have cancer." And then I went blank. But I remember, I just remember everything about that moment, and then it all went into a blur. And as you all know, it was a terrifying diagnosis and probably a word that you had never heard before. And my husband and I were like, "Cholangio what?" And we did Google. And it's not always the best thing to Google. It's terrifying. But we were told it was most likely intrahepatic cholangiocarcinoma, a rare bile duct cancer. Surgery was the only chance for a potential cure. And even with surgery, the recurrence rate was high. But at that time, and at this time still, there's no proven cure, no proven regimen. I remember my first major decision was to find a surgeon, and being from Billings, Montana, there's not a lot of experts in cholangiocarcinoma. And so we did get a second opinion with Mayo Clinic, but for personal reasons, I chose to stay home and have that surgery done there. And I had 2/3 of my liver resected. I was 41, I was a mother of 6, it was winter, it was Christmas, and I made the decision to stay home. I was really confident in my surgeons and felt like they could do the job. I had 2/3 of my liver removed. They said they had clean margins. And 15 years ago, they didn't do adjuvant therapy. I don't even remember being offered an option for adjuvant therapy. And I went home thinking, okay, maybe we're done. Unfortunately, 3 months later, at my first post-operative scan, the cancer was back in my lungs. We did a lung wedge to prove that it was the metastatic cholangiocarcinoma. And it was. And I remember at that time feeling like, okay, if I'm going to beat this, I've got to find something new. Because what was available at that time was the gemcitabine and cisplatin only. There was no other options. That was it. It was palliative care only. And I was told that maybe I could get to 2 years. And I remember just praying to God to give me 5 years to get my kids just a little bit older. I talked to my oncologist at the time into being really proactive. I told him I wanted to do a clinical trial. He actually found a clinical trial. And to this day, I have no idea what it was because I wasn't as invested as I am now. But my insurance wouldn't cover the standard of care costs that went into participating in that clinical trial. And I remember the rug being pulled out from under my feet. I didn't even know that was an option. I didn't even know that that could happen. And it was so devastating because you were presented with an opportunity that was new that maybe could have some hope, but then it was completely taken away. But for financial reasons, I didn't want to go forward with it because I didn't want to put my family in financial stress for something that may or may not work. So we went ahead and started the GemCis, and unfortunately, I'm one of those patients that has a horrible response to chemotherapy. A lot of toxicity after about 6 months, the ringing in my ears, the neuropathy in my hands and feet. I had a little bonus of steroid psychosis, which is never fun. And they took the cisplatin away. I went on gemcitabine alone, and the cancer spread from my lungs back to my liver. At that time, there was no other options available. They threw Taxotere at me, which did nothing, and my quality of life was so poor. I made the decision to stop all treatments and have whatever time I had left to have, I don't want to say quality of life for Ivan, I want to say fulfillment of life left with my family because I was spending so much time in bed. So I stopped all treatments. But what I love to tell patients is I made that decision one day, and 3 weeks later, a new opportunity was presented with the clinical trial at the National Institutes of Health, the National Cancer Institute. And I remember I was paying a medical bill online, and I quit looking for clinical trials because there was only clinical trials with different chemo combinations, and I wasn't going to do chemo. But for some reason, this trial popped up on my computer, and I read it, and I went, I think we should try this. I looked at my husband. I said, I think we should give this a shot. It has the initial chemo for a week. I knew I could get through a week. And then it's your immune system doing what it should be doing. And I said, I think we really should try this. So we took it to my oncologist, and he's like, I'll support you in whatever you want to do. But my husband and I did have to do the work. We had to make the phone calls. We had to send the records. He drew my blood at the kitchen table. We got this done. He's a nurse practitioner, but we made it go as fast as we possibly could. But I got to that point too, where I'd stopped treatment and for 3 weeks was off treatment. And when I went in for my first scan, the cancer hadn't progressed. And so I didn't qualify for this trial. And I remember going, are you kidding me? Are you kidding me? This cancer is going to progress. And so I know it's those little inclusion criteria that just really get my goat that we really have to find a way to advocate to get around. But sure enough, my husband picked up the phone and called and said, no, we're coming. And he had to later apologize to the sweet research nurse out at NCI and, apologize. But by the time we got out there 3 weeks later, the cancer had progressed. And I did enter into the trial. I want to personally thank Dr. Turcotte because he is literally responsible for me standing here today. He's the one who advocated to have the GI TIL trial at NCI. He had to sell it. He had to sell it to his colleagues to get it done. And so I truly, truly, truly owe him my life. Went in, had the initial tumors removed from my lungs. Dr. Turcotte removed them. And he harvested the TIL and grew it. And I came back about a month later to go through the treatment. To me, the worst part was the lymphodepletion, again, because I just don't do chemo well. And so that's the part that I definitely struggled the most with that first week. But it was so doable knowing that there was an end date. Like, you know, I knew I was going to get through it for a week. The next hardest part was being away from my kids for it was about 3 to 4 weeks that I ended up being in the hospital for this treatment. But I'm also so blessed because the NCI has a family lodge, and my husband was able to come and stay with me there and be with me through this entire process. The second week was the cell infusion. And again, I remember at the time, Dr. Turcotte was standing next to my bed giving me the cells, the nurse was giving me the cells, and I jokingly said to him, "Hey, are you ready to make history?" And, you know, just trying to lighten the mood, and look at what we did. Recovery, I think it was a little bit hard too. I just, you know, kind of want to be honest. The IL-2 can cause some nasty side effects. For me, it was water weight gain overnight. I think I gained around 20 pounds overnight of water retention, which is fine because then they give you Lasix and you pee it all off. But the hard part about it is you can't have a catheter because of your risk of infection. So you're getting up every 20 minutes to use the bathroom and you're just exhausted. And I know those sound like little things. Then trust me, I would do it again in a heartbeat. But I remember that just being so exhausting. And then getting your immune system enough to recover enough to be able to go home. I know I had issues with my platelets both times and ended up with quite a few transfusions. But eventually my immune system kicked in and I was able to go home. I was around the 9th person. I'm not sure on that number, but I always, for some reason, thought 9th person to enter into the trial, the first one with cholangiocarcinoma and the first one to have a response. As Dr. Turcotte said, I had almost that overall shrinkage of 30%. It was a little less than 30%. The first 6 months, things shrunk. The next 6 months, things stayed stable. And then after that, it started growing again. But this was a one-time treatment. So I had no other treatments after I went home. And I just want to go back to when I went into this trial, I had tumors all over both my lungs, and I had such a bad cough. I couldn't take 5 steps without going into a coughing fit. My kids couldn't hug me without me going into a coughing fit. And by the end of that 3 to 4 weeks in the hospital, after receiving that cell infusion, that cough went away. And I was so cautiously optimistic. I'm like, something's going on here. And I went home and for 18 months had an amazing, fulfilling life. And I didn't waste one moment. So this is some slides that I stole from Dr. Eric Tran, who also worked on my case at the NCI. I had 42.4 billion cells the first go around, with only about 25% of that TIL product targeting my ERBB2IP mutation. So these were some scans of my lungs for pre-treatment tumors, and then 7 months after that first cell infusion, and then the ones in my liver as well. So I had stabilization after that first cell treatment, but they wanted to know if we can do better. And I remember, and I lovingly call Dr. Yang. I love Dr. Yang. I absolutely love Dr. Yang out there. But my husband and I had nicknamed him Dr. Doom and Gloom when we first met him. And I don't know if that's appropriate, but that's what we kind of nicknamed him. Because I remember asking him in the beginning, okay, you know, can we do this again? And, you know, and he was just very always matter of fact and very factual and stuff. But I remember when I went, when they discovered this mutation reactive T cell and going back and the excitement that was in the air there. And then his attitude changed and I never called him that again. And to this day, I think he's, yeah, just one of the best. So again, back to the NCI for more testing to get retreated with this T cell. And I was laying in bed at NCI getting apheresis done. And Dr. Rosenberg came to the foot of my bed and his excitement about this just was so contagious to me. Like, then I was so excited. I'm like, yes, let's do this. Let's get this started. getting the courage to go back and do it a second time. I would do it a third, I would do it a fourth time, but it took a little bit of courage, and Dr. Rosenberg just made it so much easier because I'm like, okay, if he's that excited, then I'm excited too. So back for another tumor removal, they harvested the tumor again and pulled the tail from the tumor. So I got retreated with, that was the first one. And the second T cell had 95% of the cells were reactive to that ERBB2IP mutation. 126 billion T cells this time, so almost 3 times as many as the first go around. I felt like I had absolutely nothing to lose and everything to gain, because as a cholangiocarcinoma patient with not a lot of options, I think that we feel that way. I had an overall shrinkage of about 50 to 60%, 3 years of a great, fulfilling life. I'm going to change all my words, Ivan, just for you. And no other treatments, just my immune cells doing the job. But this data was published in the Science Journal, and the New York Times did an article. And after that article went out, in that first week, I received hundreds of phone calls from patients all over the world, all over the world. They just wanted to have hope. They just needed a little sliver of a possibility. They didn't need to know that this specific trial would work for them. They just needed to know that there was a possibility and an opportunity to try. These were some pictures of my lung CT after the second treatment in 2013. These lung tumors shrunk drastically. Unfortunately, in January of 2016, there started to be a glimpse of growth in those tumors again. We kind of watched things for a while, and then they decided to be proactive and go back in and remove the remaining tumors from my right lung to resequence and do the research on them. So yeah, wondering why they progressed, and that's why they took them out and they restudied them. Back to the OR, I've had four lung surgeries, and that's why I can't run 2 blocks, Dana. That's my excuse. So after that, and Dr. Turcotte, you might have to help me get through the science part, but they resequenced, restudied those tumors, and I still had the same the presence of the same mutation, and the T cells were still present in my circulation. But this time there was a presence of the PD-L1 protein. Yes, so they decided to treat me with the pembrolizumab. And so I went back in, and after the second dose of having the pembrolizumab, they did scans, and there was already drastic shrinkage. And so that opened the window for those T cells to be able to recognize the cancer again. I don't think the pembrolizumab would have worked had I not had those T cells in there to recognize the cancer. And that is the last treatment I have had in 8 years. 8 years ago in March since I've had any treatment for cholangiocarcinoma. And everyone always asks me if I'm in remission. And I love to hear Dr. Rosenberg say she's cured, and I'm going to go with what he says, but I'm always like knocking on wood, and I don't care what's going on in there as long as it's not growing and as long as there's no evidence of disease, and I get to live every day to the fullest. Again, some other scans of my lungs from the after receiving the pembro doses. So there was a lot of highs and lows, right? I was so excited to find that trial and just say, okay, there's an opportunity that didn't involve chemos that were making me miserable. The other high about it is it happened to be at the NCI, the National Cancer Institute, which is a federally funded facility. And now there's chaos going on in the world that I live in. But the only reason I could enter into this trial is because the cost of all the medical costs were covered, and they covered my costs from flying from Montana to Maryland. And as a mother of 6 and having a big family, there's no way we could have afforded to have done this. So if I hadn't had that opportunity, this wouldn't have happened for me. The surgeries were, part of the process. And I would do it again. I feel as cancer patients, most of us just want the surgeons to cut it out to begin with, just get rid of it as fast as possible. So the surgeries were highs and lows, a lot of scars. Was it working? Was it not working? That 3 years of up and downs was hard, but doable. The magic of NCI, like I said, what an incredible facility. Walking into Building 10, from the minute you get on campus, from the security guards to every single person, from the custodian who would clean my room at night so I wouldn't get an infection, who would also stop by and say a prayer with me by my bed. Like, it was just amazing. The nurses, the researchers were wonderful. Dr. Tran used to come down out of his office every time I would come for an appointment and explain to me what was going on in words that I could understand. But I was also away from my family. During my second TIL treatment, I was invited to speak at a cancer support community event, and I couldn't do it because they're like, you need to come now. And I'm like, okay, I'm there. So my daughter at the time, I think was maybe 2021, and she said, Mom, I'll do it for you. And she went and spoke and told my story. And I remember watching it in the hospital when I was at NCI and just being so proud of my kids and the strength that they had going through this as well. The bonus was my niece and nephew lived close to NCI, so I got to watch them grow up from this age to now they're grown teenagers. And yeah, you lose your hair. I lost my hair 3 times, but it grows back. I wanted to find a way to pay it forward because I knew how fortunate I was to be here. And so I started volunteering with the Cholangiocarcinoma Foundation. I came on as a moderator on the discussion board, and there was patients from all over the world. 15 years ago, there was just really a discussion board. That's all the foundation was. That's how they started. It was just trying to make connections around the world so that patients could share resources and information. And I just wanted to help patients navigate it. And now I'm happy to say that about 8 years ago, I came on as a full-time staff, and I've had the opportunity to share my experiences and use my voice to represent all the patients on many national panels, some at the NCI, on the NCCN hepatobiliary guidelines, and I'm so privileged to be a part of the C3 collaborative. It definitely has brought it full circle for me to start as a patient, to now be a patient advocate and be able to help other patients, to go to these big cancer conferences at AACR or ASCO and be sitting in the audience, and then all of a sudden this slide pops up and I'll be like, pinch me, because it's so unreal for me to do that. And then for C3 to start and to be able to be the patient advocate on it instead of the patient participating in it has just brought everything full circle. I am so unbelievably grateful for my NCI team and for all the researchers who are doing research to advance the treatments for our cholangiocarcinoma patients. Also very, very, very grateful to be a part of the CCA community. I think as all of you guys discovered yesterday, it's never a community you choose to be a part of, but it happens to be one of the most amazing communities I have ever participated in or been a part of. I don't know why the nicest people get this disease, but they do. And it's just so great to see the connections that are being made and the collaborations that are being made and the hope that's in this room. And I do want you to repeat that survey wherever Samara is. Yes. So thank you. And I appreciate the opportunity to be here.